Welcome back, fellow scientists. All right, so we are still in chapter 10. We're moving on from white blood cells and platelets and kind of our overview that we did last time into red blood cells. So as you can see in OneNote, I have put a blank copy under Content Library, blank notes, and then I also pushed it out to your notebook under handouts. So I was encouraged actually as I was doing that. I saw that a couple people actually took notes in their on the blank notes that I provided in their handouts. So good job. Keep up the good work. Way to be good students. All right. So we're just going to dive right in because we have a lot of stuff to do today, and I don't want to make a video that's too long. Excuse me. So I also noticed that on the last video, Shaylin commented that I should use mustard yellow for the next color. So that's the closest mustard yellow that I could figure out. So we will go ahead and honor that, and we'll go ahead and write down. Today we are talking about red blood cells. Red blood cells, and specifically we are talking about the A, B, O, and RH blood groups. So a little bit of background. You remember all the way back from freshman year, maybe some of you that have taken AP Bio, you'll remember that this is genetically determined. So I want to go back and talk about that. Genetics. So if you have type A blood, it could be either I to the A, I to the A, or I to the A, little I, right? If you have type B blood, you could be I to the B, I to the B, or I to the B little I, right? So this would be the homozygous condition. This would be the heterozygous condition. If you have type O blood, you only have one choice. You can be little I, and that's it. And then the Rh factor is determined by something different. If you are Rh, let's see, Rh positive, right? You could have Rh plus, Rh plus, or you could have Rh plus, Rh minus. So again, heterozygous condition, or sorry, homozygous condition rather, heterozygous condition. If you are Rh negative, you can only have Rh negative, Rh negative, right? So all that genetic stuff is coming back that everyone has at least two alleles for every gene, right? One allele you got from your mom, one allele you got from your dad. And then when you're making the next generation, then you can only pass on one allele to your offspring. So if you're type A but you're heterozygous, you could either pass on the A allele or the little I allele. And so all sorts of different genetic problems, like if both parents are type A, could they have a child that's type O, right? If there's a AB parent, and if then there's just a regular B parent, then what blood types could we see? If there's two parents that are both type O, then could they produce a child that's type B or whatever? So, anyways, that's neither here nor there. This isn't a genetics course. This is an anatomy physics course. But I thought I'd mention that because that ties in with what we've learned before. Uh, what's so that's neither here nor there, like I said. But what is important, not that that's important, but what is important are these two words right here. So antigen, right, and antibody. All right, so antigen, we're going to go ahead and define this. Antigen is a cell surface protein used for identification. Cell surface protein, protein, yeah, protein used to ID a cell. So that's an antigen, and you can see that they come in different shapes, and all of you hopefully remember from biology and AP biology that, right, shape determines function. Okay, good. And then antibodies are immune system proteins. They're not cells. They're immune system proteins So immune system proteins that help with the immune response. So if there's an antibody that attaches to a certain cell, that's a bad thing for the cell, usually, because then that's going to create an immune response. So immune system proteins that like identify the bad guys, or they help with the immune response. I'm not sure why the computer does the writing worse as it goes, but it does. So I apologize. Immune system proteins help with that's an H. Help with immune response. If an antibody attaches to a cell, that's a bad thing for the cell. So it helps to identify non-self proteins. And again, they come in different shapes. Right here, we can see kind of a semicircle one. Here we can see kind of a triangle one. Here's another semicircle one. Right. Um, well, that doesn't. I guess all these are semicircles. Anyways, maybe that's a bad diagram, but regardless, you get the point. Shape determines function. The antibody has to be the right shape to attach to the shape of the antigen of the cell surface protein. All right, now how does this play into red blood cells? So red blood cells have antigens, right, on their cell surface. So if you are type A, then you have type A antigens and you have anti-B antibodies. You have non-self antibodies. Notice that they are in the plasma, right? And then these are actually attached to the cell. Okay, if you are type B blood, you have B antigens, and then you have antibodies that will identify type A because it's non-self, right? You'll identify type A and then mount an immune response against type A. If you have AB blood, then you have type A antigens and type B antigens on your cells, and so your body doesn't make any antibodies against those because this is self, right? And otherwise that would be a bad thing. That would be incompatible with life. And then if you are type O blood, you have no antigens on your cell. So your immune system makes both antibodies to identify as non-self and to again mount an immune response and kill it, right? So for instance, if person who is type B, right, was infused with blood, was given blood that is AB, okay, like that, would this person right here, would this type B person, would their immune system mount an immune response? Yeah, because they have the anti-A, and this blood cell, this AB blood cell, has the A antigen, right? And so these would attach to that, and it would clot up, and not clot because there's not platelets enough, but it would form clumps, right, of red blood cells, and those clumps of red blood cells, they would get stuck in the small little capillaries in between the arteries and the veins. And then those cells would lyse, and then the hemoglobin would be released into the bloodstream, and then that really affects the kidneys because the kidneys are kind of the filtering thing to get out all the bad stuff. And so most people who have a transfusion reaction, they actually die from kidney failure, right? They can they can kind of survive, and we can get them the right blood and different things like that. But if the clots affect the kidneys and if the kidneys start shutting down, then that again is incompatible with life, and that just doesn't work. So I'm going to be intentionally vague, and you'll find out why at the end. But that's just one example to kind of get the point across of antigens versus antibodies and what people normally have. So now this next slide talks about the Rh factor. Right, so Rh factor is named so because it was first discovered in the rhesus monkeys, and then it was later discovered in humans. So basically, if you're Rh positive, you have the protein, right? And then if you're Rh negative, you don't have the protein. You don't have the antigen, the cell surface protein, right? So if we put that together with our A, B, and O blood types, you'll notice that if a person is AB positive, they have all of the cell surface proteins, and so they're going to make no antibodies, right? Because if they did, then the antibodies are attached to self, and that would be bad, right? On the other end of the spectrum, if you are O negative, you'll notice that you have no cell surface proteins, and so you're going to make all the antibodies, right? All of those little Y-shaped things that are going to attach and they're going to cause them to clump and different things like that. So you would have an antibody, you'd have an A antibody, right? We'll just use the same kind of shape that they use right up here, right? And you would have a B antibody. That's supposed to be a little bit more angular. That's supposed to be a little bit more circled. Sorry. And you would have an Rh antibody, right? Because you don't have any of those. And so if there are any of those proteins in your bloodstream, that's non-self. And so your immune system needs to identify that and it thinks that it needs to attack that and deal with that. All right, so that's bringing in the Rh factor. And that's actually all that I wanted to teach you for today. We'll go into hemoglobin and sickle cell anemia and blood doping and erythropoietin in the next video. Coming up on, when would that be? Tuesday, probably. But like I said, I have been intentionally vague about the most common type of blood type or the transfusion reactions, universal donor, universal recipient. An interesting thing to think about, like we talked about with the genetics before, is if you are type A and the genetics work out such that you're pregnant with a type B baby, does that have any ramifications? Like, is that possible, or what happens? You can probably think up some other questions. So I want to try a discussion board assignment. So I'm going to post a discussion board assignment on LMS RunWeb, and then your homework for today is to ask a question and then answer a question. Answer someone else's question. Don't answer your own question. So ask a question, answer a question, and then that'll be your assignment today. So this has been Mr. Leo. Today we've talked about the ABO and RH blood groups. Hopefully it has been fun and informative. And hopefully once coronavirus dies down, I will see all of you on the flip side.